here we are in my backyard today, and the reason why we're here is because I want to talk about the pressure flow hypothesis for how plants are going to be moving sugars from the source to the sink. And what I really like about the crocuses over here in our ginkgo tree is that both of these plants are going to be showing us the pressure flow hypothesis, but they're going to be moving the sugars in opposite directions. So first I want to start with the crocuses. And what's happening right now with the crocuses is they are doing photosynthesis. So here in the leaves, we have tons of photosynthesis happening. They're doing the light reactions and the dark reactions. They're eating carbon dioxide and making glucose. Now they're going to be moving that glucose from the source, these leaves, down into the corm, into that stem, which is going to be the sink. In order to do this, companion cells are going to actively pump the sugars from the source cells into the sieve tube members, part of that phloem tissue. When we get lots of sugars in the phloem tissue, water is going to move from the xylem into the phloem, and it's going to push those sugars down toward the sink cells. So in our crocus stem, remember, is modified. It's going to be called a corm. It is going to be underground. It's going to be storing those sugars. So the sugars move through the sieve tube members down to the companion cells at the sink. The companion cells at the sink are going to actively pump those sugars into storage cells like parenchyma cells. Those glucose molecules will get linked together into starches, and the starches will most likely get stored in an amyloplast organelle in those parenchyma ground tissue cells. So for the next few months, our crocuses are going to be doing lots of photosynthesis, pushing those sugars using the pressure flow hypothesis, pushing the sugars through the phloem, storing those sugars in the corm. By the middle of June, all of the leaves from my crocuses will be gone. All that will be left are these little nuggets, these little corms in the soil. The sugars that they store in the next couple of months have to get them through the rest of the summer when the crocuses are dormant, and then get them through the winter into the next spring season, where they'll use those sugars to make new leaves, put up hopefully a beautiful new flower, and then start the whole process over again. So our crocuses are moving sugars from source to sink, and it's moving in the more traditional manner, the way how we see in a lot of those videos that came along with the lecture slides. Our ginkgo tree over here is also using the pressure flow hypothesis, but it's using it in the opposite direction. Last summer, the leaves were photosynthesizing, and a lot of those sugars got stored down in the roots. Now what's happening right now is those sugars are being moved up through the body of our ginkgo tree. So companion cells down at the source, down in our roots, are actively pumping the sugars into the sieve tube members. We get a hypertonic solution in our sieve tube member. Water moves from the xylem into our sieve tube member and pushes those sugars upwards, pushes the sugars up against gravity. That's how much pressure there is when water is flowing into our sieve tube member. Those sugars are flowing up through the trunk, flowing out to the branches, and they're going to end up here in the little nodes. At the nodes, we have companion cells actively pumping the sugars out of the sieve tube members and into what are the developing leaves. Essentially what our ginkgo tree is doing is it's locked in this race. Does it have enough sugar down here in the roots stored from last year to be able to start this process of growing the leaves? Can it get those leaves out soon enough to start photosynthesizing and making sugars? Once it gets those leaves out, we're in the next round, the next race to photosynthesize and make enough sugars to store back in the roots to replace what was used this spring, help get this tree through the winter, and start the whole process over again next year. I hope these two plants help you to understand the pressure flow hypothesis. If you have any questions, please email me.